Now that we've covered the basic physiology of the cardiovascular system, we need to sort of switch gears a little bit and talk about how the cardiovascular system responds to the stresses of exercise, particularly the increased oxygen demand of active skeletal muscles during exercise. In other words, during exercise and as muscle contractile activity and intensity increases, there is obviously an increase in the need for oxygen for the muscle to produce ATP aerobically with. And thus, the cardiovascular system must respond to meet that demand by essentially distributing more oxygenated blood to active muscles during exercise. This means the heart, as well as the arteries, need to undergo immediate changes for that to happen. So in the next section of this lecture series, we will talk about how the heart and arteries go about getting more oxygenated blood to the active muscles during exercise, ultimately to support aerobic ATP production. First, let's discuss how the heart responds to exercise. But before we do that, we need to understand key cardiac variables, heart variables in other words. We generally understand that when we exercise, our heart rate increases. Heart rate is easily measurable and observable, so this is a well-known fact. But other than heart rate, there are other variables associated with the heart that undergo changes for the ultimate purpose of getting more blood flow to the active muscles. And please keep in mind that the changes in the heart during exercise is only one piece of the puzzle, because there are also changes that occur in the vasculature, which we'll discuss later on. So first we have heart rate, which is the number of times the heart beats per minute. So this is the number of times the atrium and ventricles contract per minute. Remember, one heartbeat is an atrial contraction followed by a ventricular contraction. Now, I want to emphasize one key aspect of heart rate, and that it is a measure of rate, meaning it is something per unit of time. This is going to be important for you to remember as we discuss the most important cardiac variable, which is cardiac output, which I will define in a minute. The next key cardiac variable is stroke volume, which is something that we can't measure easily, at least compared to heart rate. Stroke volume is something that can be estimated for an individual, but it is very difficult to directly measure. Stroke volume is the volume of blood ejected from the left ventricle per beat or per ventricular contraction. So remember, the left ventricle, when it contracts, ejects blood into the aorta and then to the systemic arteries, which carry oxygenated blood to the rest of the body. So heart rate is a measure of heartbeats per minute, while stroke volume is the volume of blood ejected per beat. So what do we get when you multiply these two variables together? You get cardiac output, which is denoted by the letter Q. Cardiac output is the volume of blood pumped out by the heart per minute. So based on that definition, you should be able to understand that Q equals heart rate times stroke volume. This relationship is very important for you to understand, especially when it comes to understanding the heart's response to exercise. So the two factors that contribute to cardiac output is heart rate and stroke volume. So hypothetically, if heart rate increased and stroke volume remained the same, how would cardiac output be affected? It would obviously increase according to this relationship or formula. What if heart rate increased and stroke volume increased? Again, according to this simple formula, cardiac output would increase. If, say, heart rate decreased and stroke volume remained the same, cardiac output would decrease. So whatever changes occur in heart rate and/or stroke volume, cardiac output would be affected. Now, if you look at the definition of cardiac output, it is also a rate, which again means something per unit of time. So essentially, cardiac output is the rate of blood volume being pumped out of the heart into the arterial system and to the rest of the body. In other words, it is simply a measure of blood flow from the heart. So as far as units of measurements are concerned, heart rate is beats per minute, stroke volume is in liters or milliliters, and cardiac output is liters or milliliters per minute. Please keep that in mind. So first, let's see how heart rate responds to exercise, more specifically to exercise intensity. Now, this one is a no-brainer because most of us understand how heart rate responds as you increase intensity of exercise. So we won't spend too much time on here. On the x-axis, you see a scale of intensity as a percentage of Vo2 max, which again is maximum oxygen consumption. Vo2 max occurs with maximum intensity, and as I emphasized in the prior lecture series, it reflects the maximum capacity for aerobic ATP production. So 100% VO2 max is maximum intensity, and any percentage below that will be submaximal intensity. Now on the y-axis, you have heart rate in beats per minute. And like most of us understand, as intensity increases, heart rate increases in a proportionate fashion. And maximum heart rate, or heart rate max, is achieved at 100% VO2 max intensity, or maximum intensity. Just for some important terminology, however, anything below maximum heart rate, as you see here, is simply referred to as submaximal heart rate. So I might ask a very simple question, such as, during what situation is maximum heart rate observed? The potential answers would be at 100% VO2 max, or simply at VO2 max or maximum intensity. So what about stroke volume? The relationship between stroke volume and exercise intensity is a little bit more complex than heart rate. Now, just as a reminder, stroke volume is the volume of blood ejected from the heart, more specifically the left ventricle, per beat or contraction. So again, we have exercise intensity represented as a percent of VO2 max on the x-axis, and we have stroke volume on the y-axis in the units of milliliters per beat, or left ventricular contraction. Now, just like heart rate, stroke volume increases proportionately to exercise intensity, as you see here. But the difference is it increases proportionally to exercise intensity only up to about 50% VO2 max intensity, which is considered about moderate intensity. And you can see that here in the graph as the red dotted line, which represents stroke volume, and maxes out at around 50% VO2 max intensity. This means stroke volume does not increase any further as intensity increases past 50% VO2 max intensity. So if I ask the question, how does stroke volume change from rest to about 50% VO2 max intensity, the answer would be it increases proportionately to intensity, meaning as intensity increases, stroke volume increases. And this is again only from rest to about 50% VO2 max intensity. And if I ask the question, how does stroke volume change at exercise intensities of 50% VO2 max and above? The answer would be stroke volume does not change because it has already reached its maximum at about 50% VO2 max intensity. I could also pose this question differently, such as, stroke volume increases proportionate to exercise intensity until blank. The answer would be 50% VO2 max intensity. So the next question is, why does stroke volume max out at around 50% VO2 max intensity? And why can't it increase any further from here, even with an increase in exercise intensity? Well, first we need to understand the factors that determine stroke volume. The two factors of stroke volume is end diastolic volume and end systolic volume, or EDV and ESV for short, respectively. End diastolic volume is the volume of blood that fills the ventricle before contraction, aka systole. And the end systolic volume is the residual amount of blood left in the ventricle after contraction, or again, systole. So if stroke volume is increasing, what changes are occurring with end diastolic and end systolic volume? First, we have an increase in end diastolic volume, meaning there is increased amount of blood in the ventricles before it contracts. This means in between each ventricular contraction or heartbeat, there is more blood filling the ventricle. And during exercise, end diastolic volume increases because there is greater venous return. Why? Remember, during exercise, the muscle pump mechanism is highly active, and that will facilitate blood returning to the heart via the veins, aka venous return. This means that there is more volume of blood filling the ventricle between each contraction or heartbeat. So stroke volume increases partly because end diastolic volume increases. Stroke volume also increases because there is a decrease in end systolic volume, which means that there is a decrease in the amount of blood left in the ventricle after it contracts, Again, this is the volume of blood left in the ventricle after it contracts. And again, stroke volume is simply the amount of blood that was ejected from the left ventricle after it contracts. So if you think about the description of stroke volume, end diastolic volume and end systolic volume, you would know that stroke volume equals end diastolic volume minus end systolic volume. So if end diastolic volume increases, as we just said, and end systolic volume decreases, as we just said, according to this formulation, stroke volume would increase. And this is what explains the increase in stroke volume from rest to about 50% Vo2 max intensity. So let's now discuss why end diastolic volume increases and end systolic volume decreases within this intensity range up to 50% Vo2 max. So as I mentioned earlier, end diastolic volume increases mainly because there is more venous return due to the active muscle pumps essentially pushing blood through the veins back to the heart. This means more blood is being essentially dumped into the ventricles in between contractions. So how does the ventricles filling up with more blood between contractions cause an increase in stroke volume? Now this explanation is based on the Frank-Starling law, which states that there is a direct relationship between the force of ventricular contraction and the resting length of the ventricle myocardium. Let me explain. The resting length of the ventricle myocardium is essentially referring to the stretch of the ventricle. So when the ventricle is filled up with more blood, there is more stretch of the ventricle. Let me repeat that. So when the ventricle is filled up with more blood, there's more stretch of the ventricle. It expands more because more blood is filling it. And because of this stretch of the ventricle, more specifically the ventricular myocardium, there is going to be a more forceful ventricular contraction, thus causing greater volume of blood ejected, i.e. stroke volume. So to lay it out in a simple visual, you can see here that with an increase in end diastolic volume, which means that there's more blood filling the ventricle, this would then lead to an increased stretch of the ventricular myocardium, which leads to a greater force of ventricular contraction, causing more blood to be ejected from the left ventricle, aka increased stroke volume. And how does this affect end systolic volume? It would decrease it because there's less blood left over in the ventricle after it contracts. And this is again because the ventricular contraction was more forceful. So if I asked why does end diastolic volume increase from rest to about 50% Vo2 max intensity, the answer is because of increased muscle pump activity and increased venous return. If I ask why does stroke volume increase from rest to about 50% Vo2 max intensity, one of the proper answer choices would be because of a greater ventricular contraction due to greater stretch of the ventricle. And again, what causes this increased stretch of the ventricle? It is the increased filling of the ventricle with blood, aka increased end diastolic volume. Now, if I asked which of the following causes a decrease in end systolic volume from rest to about 50% Vo2 max, the answer is increased volume of blood ejected from the ventricle following contraction, which is the same thing as saying increased stroke volume, thus leaving less blood left in the ventricle after contraction. Now also keep in mind when I'm referring to the ventricle here, I'm always talking specifically about the left ventricle, which again ejects blood into the systemic arteries to the rest of the body's tissues. So now let's go back to the question of why does stroke volume plateau starting at about 50% Vo2 max intensity? So we know why stroke volume increases from rest to 50% Vo2 max intensity, which was discussed in the previous slide. But why does it max out at 50% Vo2 max? And why can't it increase any further with intensities increasing above 50% Vo2 max? Now this has to do with heart rate continuing to increase at 50% Vo2 max intensities and above. At 50% Vo2 max intensity and above, the heart rate has increased to the point where the time available between contractions of the ventricle is reduced. If the time between contractions is reduced, then there is a decreased time to fill the ventricle with blood between contractions. So according to the Frank-Starling law, this means that due to decreased ventricular filling time, there cannot be any further increases in end diastolic volume, and therefore no further increase in ventricular stretch, and therefore the force of ventricular contractions cannot increase any further, and therefore stroke volume cannot increase any further, and thus maxes out. Now, just for clarification, these down arrows here for EDV, stretch, and force do not necessarily mean that they decrease. It just means it cannot increase any further at a 50% VO2 max intensity and above. And the same goes with this up arrow for ESV. It just means that ESV cannot decrease any further. And thus, according to the simple formula for stroke volume, if EDV cannot increase any further and ESV cannot decrease any further, there will be no further change in stroke volume. So the ultimate reason why maximum stroke volume is observed at around 50% VO2 max intensity is because of the reduced ventricular filling time caused by the increasing heart rate, as you see in this figure right here. You can see in this figure for heart rate that at 50% VO2 max intensities and above, heart rate continues to climb in a proportionate fashion. So with this reduced ventricular filling time, this would cause no further increases in end diastolic volume and no further increases in ventricular stretch, and thus no further increase in ventricular force. And this is why stroke volume at around 50% VO2 max intensity is already maxed out. Okay, so we see here that heart rate increases from rest to 100% VO2 max intensity where it maxes out. Stroke volume increases from rest to about 50% VO2 max where it maxes out. And from 50% VO2 max intensities up to 100% VO2 max intensities, stroke volume does not exhibit further increases. It's already maxed out. Again, why? Because the heart rate is so fast that there is reduced ventricular filling time. Thus, EDV, stretch, and therefore force cannot increase any further at this point right here. And thus stroke volume cannot increase any further at this point right here. Okay, so now what about cardiac output, or Q for short? Remember, Q equals heart rate times stroke volume. Therefore, cardiac output is the amount of blood ejected from the heart every minute. So first, let's see how cardiac output changes from rest to about 50% VO2 max. As you can see, it increases proportionally to exercise intensity. Now, why does cardiac output increase from rest to 50% VO2 max intensity? Well, let's take a look at the factors that contribute to cardiac output, which again are heart rate and stroke volume, HR times SV. Remember, from rest to 50% VO2 max intensity, heart rate increases proportionally. And also from rest to 50% VO2 max, we know that stroke volume increases proportionally. So if cardiac output equals heart rate times stroke volume, how would cardiac output change from rest to 50% VO2 max intensity? It would obviously increase because the two factors that contribute to cardiac output increases. Again, Q equals heart rate times stroke volume. So if heart rate and stroke volume increase, obviously Q would increase as well. And this is the case from rest to about 50% VO2 max intensity. Now here's where it gets a little bit tricky. What about changes in cardiac output from 50% VO2 max all the way up to 100% VO2 max intensity? Again, let's take a look at the factors that contribute to cardiac output. We know that from 50% VO2 max to 100% VO2 max intensity, heart rate continues to rise. But what about stroke volume? We know that stroke volume at 50% VO2 max intensity has already maxed out. So from 50% VO2 max up to 100% VO2 max intensity, there is no further change in stroke volume, as we discussed in the previous slide. So the only contributor to an increase in cardiac output from 50% VO2 max intensity and above is heart rate. Since again, stroke volume is already maxed out. And this is why cardiac output increases from 50% to 100% VO2 max intensities, but there is a slight plateauing of this increase, as you can see in this figure right here. And going back to the formula, it is because instead of two factors contributing to the cardiac output increase, there is now only one factor contributing to an increase in cardiac output, and that is an increase in heart rate, not stroke volume. So if I ask the question, which of the following factors contribute to an increase in cardiac output from 50% VO2 max intensities to 100% VO2 max intensities, the answer would be an increase in heart rate, and that's it. If I asked which of the following factors contribute to an increase in cardiac output from rest to 50% VO2 max intensity, the answer would be both an increase in heart rate and an increase in stroke volume. So now let's talk about blood flow to the peripheral tissues, particularly oxygenated blood flow through the systemic arteries. Again, this is blood leaving the left ventricle to the rest of the body. Now, generally speaking, you can consider cardiac output, or Q, simply as blood flow. So this table demonstrates the amount of blood flow distributed to various tissues in the body as a percentage of total cardiac output and the actual cardiac output in liters per minute. We have to understand generally during exercise, skeletal muscle, particularly the ones that are contracting during exercise, require more oxygen to support aerobic ATP production. Therefore, active muscles during exercise require greater distribution of the arterial/oxygenated blood, and thus a greater distribution of the total cardiac output. So let's go ahead and look at oxygenated blood flow through the arterial system as a percentage of total cardiac output. At rest, skeletal muscle receives about 20% of the blood flow. The heart itself receives about 4% of the blood flow. The brain receives about 14%, 27% for the liver, kidneys 22%, skin 6%, and other tissues 7% of the total cardiac output. Now let's look at how this distribution of oxygenated arterial blood changes from rest all the way up to maximum intensity exercise, so at 100% VO2 max intensity. As you can see, as a percentage of the total cardiac output, there's heavy redistribution of the cardiac output towards skeletal muscle. The muscle at maximum intensity receives about 84% of total blood flow or cardiac output. And the other tissues, which are less active during exercise than skeletal muscle, receive a lower percentage of the cardiac output. Well, why? Well, as I indicated earlier, it's because the contracting skeletal muscles during exercise require more oxygen for ATP production aerobically, and thus require more arterial/oxygenated blood. As you can see, as a percentage of total blood flow or cardiac output, arterial blood is redirected away from less active tissues during exercise and towards active skeletal muscles. I will repeat, as a percentage of total blood flow or cardiac output, arterial/oxygenated blood is redirected away from less active tissues during exercise and toward active skeletal muscles, and that is denoted here. Now, if you look at this from an actual cardiac output perspective in liters per minute, you can clearly see that muscles receive increasing arterial blood flow from rest to maximum intensity exercise. And this should make sense to you as to why. Again, it is because contracting muscles require more oxygen via arterial blood flow with increasing exercise intensity to support, again, aerobic ATP production. From rest to maximum intensity exercise, the cardiac output, which again is oxygenated arterial blood, increases from 1 liter per minute to about 21 liters per minute for skeletal muscles, more specifically the active skeletal muscles during exercise. So when you look at all the tissues during exercise, and this is again at the maximum end, where would you say that blood flow is redirected to? It is redirected or redistributed towards active muscle tissue, contracting muscle tissue. So muscle, more specifically the active muscles, get more of the arterial/oxygenated blood flow during exercise compared to other less active tissues. Now, as you can see in this figure, total cardiac output during maximum intensity exercise is about 25 liters per minute. So this is maximum cardiac output. And this is achieved during, again, maximum intensity exercise. And if you take roughly 84% of this figure, which we said was the percentage of cardiac output that is distributed to active muscles during maximum intensity exercise, this equates to roughly about 20 to 21 liters per minute, as indicated in the previous slide. At rest, the total cardiac output is about 5 liters per minute, meaning your heart pumps out 5 liters per minute at rest. And at rest, the muscle receives about 20% of that cardiac output, which equates to about 1 liter per minute, which again was emphasized in the previous slide. So to summarize, as exercise intensity increases, there is greater arterial blood flow to active muscles. More specifically, the arterial system redirects blood flow away from less active tissues to more active tissues during exercise, i.e., contracting muscles. So we know that with increasing exercise intensity, cardiac output increases, meaning your heart pumps out more blood per minute. But the question here is now, how does the arterial system redistribute that increasing cardiac output to the areas that need it the most during exercise, the active skeletal muscles? In other words, how does the arterial system redirect oxygenated blood to contracting skeletal muscles during exercise that need it? To answer this question, let's first discuss the vascular response to exercise, more specifically, the arterial response to exercise. So what happens in the arterial vessels during exercise? Now pay careful attention here because this has been an area of confusion for many students in the past. During exercise, we establish the fact that sympathetic activity increases. It's a fight or flight response. We know that the sympathetic activity is going to increase the activity of the heart. And this is responsible for the increase in cardiac output, which again is the volume of blood ejected from the left ventricle into the arteries per minute. So the question now is, how does increased sympathetic activity during exercise affect arteries? Generally speaking, arteries, more specifically their smooth muscles, have the ability to contract or dilate, which is termed vasoconstriction and vasodilation, respectively. Increased sympathetic activity, like during exercise, stimulates vasoconstriction of all arteries. I will say that one more time. Increased sympathetic activity, like during exercise, stimulates vasoconstriction of all arteries in the body. This occurs through the stimulation of smooth muscle contraction, and that is shown here in this simple figure. Now here, this is a cross-section of an artery, and the lighter pink outer wall represents smooth muscle. The circle inside represents the lumen, which is where blood flows. At rest, you have passive vasodilation, mainly because there's no stimulus for contraction or vasoconstriction. But as you exercise, there's increased sympathetic activity, which, as I said earlier, causes vasoconstriction, and that is represented here in the figure to the right. As you can see, the smooth muscle wall is in a contracted state, causing a narrowing of the lumen or the blood passageway. So what does this vasoconstriction from the sympathetic activity ultimately result in? It causes vascular resistance to blood flow because the passageway is now narrowed. But the more important effect of vasoconstriction due to sympathetic activity is that it causes high pressure in the constricted arteries. This is the same concept of squeezing a toothpaste tube, creating high pressure on the part of the tube that you are squeezing. Now hold on to this thought for a minute. We will get to why this is important. So then how does your body then increase blood flow to active muscles during exercise when sympathetic activity is causing vasoconstriction and a more narrow pathway for blood to flow? There is increased blood flow to active muscles during exercise because arterial vessels specifically surrounding active muscles undergo vasodilation, while all other arteries are again undergoing vasoconstriction due to the sympathetic activity. So as you can imagine, this creates a high to low pressure gradient, and blood will flow from high to low pressure from arteries that are constricted to arteries that are dilated, such as the one, again, surrounding active muscles. Now the question is, how does this occur? Now, many of you may believe that because sympathetic activity causes vasoconstriction, the vasodilation surrounding active muscles during exercise is caused by parasympathetic activity. This is not the case. Vasodilation of arterial vessels surrounding active muscles during exercise is not, I repeat, not caused by increased parasympathetic activity just on those vessels. There is no practical situation where sympathetic activity and parasympathetic activity increase simultaneously. As discussed earlier, when sympathetic activity increases, parasympathetic activity decreases. So there is no way that the dilation of the arterial vessels surrounding active muscles during exercise is caused by targeted parasympathetic effects on those particular vessels. So what causes vasodilation of arteries surrounding active muscles during exercise when all arterial vessels are under sympathetic influence? The proposed mechanism is called sympatholysis. Now, if you break this term down, remember lysis means breakdown, and sympatho is referring to the sympathetic influence. So sympatholysis is referring to the breakdown of sympathetic influence. In other words, the sympathetic activity specifically on the arterial vessels surrounding active muscles is blocked, or more specifically, overridden. And thus the opposite effect of sympathetic activity on the arterial vessels will occur, which is vasodilation. Now here is a figure to illustrate this entire process. Now just imagine you have a main hose and it branches off into two separate hoses. Then you turn on the faucet and the water flows. The main hose and the two branched off hoses are of same diameter, so water flows equally through all the hoses. Say you constrict the main hose and just one of the hose branches, and you leave the other hose branch alone. Where will the water flow? It will go towards the path of least resistance. In other words, it will go from high to low pressure, from where the hose is constricted to where it's not constricted. I'm sure all of you can visualize this pretty easily. Well, this is how the arteries respond to get more blood flow to active muscles during exercise. So here we have a very simple figure of arteries. You have a main arterial vessel here, and we have arterial vessels leading to less active tissues during exercise, like the gut, and we have arterial vessels leading to active muscles during exercise. Now, as I said earlier, during exercise, there's increased sympathetic activity, which causes vasoconstriction in all arteries. Again, sympathetic activity causes vasoconstriction in all arteries. However, the sympathetic influence on arteries surrounding specifically active muscle tissue is broken down or overridden through a process called sympatholysis. And those specific arteries around active muscle tissue undergo vasodilation. This creates a high to low pressure gradient. The high pressure is in arterial vessels that are undergoing vasoconstriction and not undergoing sympatholysis. And the low pressure is in the arterial vessels surrounding active muscles that are dilated due to sympatholysis, overriding the sympathetic influence on just these muscle arteries. So if you just look at this and just imagine it, you will know that blood will flow towards the path of least resistance, from high to low pressure, from here to here. So blood flow will rather favor this pathway as opposed to this pathway. This is how more blood flow or cardiac output is redirected towards active muscle during exercise. This is just commonly referred to as shunting. You're shunting blood away from less active tissues to more active tissues that require more oxygenated blood. And again, this is the case for contracting muscles during exercise. Now, without this arterial response as illustrated here, active muscles will not receive more blood flow and thus more oxygen delivery. Also, as you can imagine, having healthy, well-functioning arteries is an important factor for optimum performance and fatigue resistance. Again, if arterial blood flow is limited to active muscles, then oxygen supply will be limited to active muscles, which will reduce aerobic ATP production, which is reflected by low anaerobic threshold and low aerobic capacity. Okay, so we know that this high to low pressure gradient is caused by the specific dilation of arterial vessels surrounding active muscles during exercise. And I indicated this is not caused by parasympathetic activity just on these vessels here. This vessel is still under sympathetic influence, but these vessels are undergoing a process that overrides that sympathetic influence, and this is referred to as, again, sympatholysis. This is very important for you to understand, and again, it is an area of confusion for many students in the past, so please spend some time on this and understand it before moving forward. So if I ask the question, which of the following effects does the sympathetic activity have on arterial vessels during exercise? The answer is vasoconstriction. This is directly indicated in slide number 36. Many students indicate vasodilation as the answer to this question, but that is incorrect. They answer vasodilation because they are thinking about the vasodilation of the arterial vessels surrounding active muscles. Remember, the arterial vessels surrounding active muscles are still under sympathetic influence and they want to constrict, but through sympatholysis, which we will discuss in the next slide, the sympathetic influence on those arterial vessels surrounding active muscle is overridden and essentially opposed, and thus only those specific arteries undergo vasodilation, thus creating a high to low pressure situation in the entire arterial system, allowing for more blood flow distribution towards the active muscles. Now, hypothetically, if sympathetic activity caused vasodilation of all the arteries in the body during exercise, we would be at a very high risk of dying due to extremely low blood pressure, and there would be no way for muscles to get more blood flow. Also, if I ask the question, how do arterial vessels surrounding active muscles during exercise vasodilate? The answer would be sympatholysis, or any of the mechanisms underlying sympatholysis, which will be discussed next. Again, it is not due to parasympathetic activity specifically on those muscle arterial vessels. That is not how vasodilation in those vessels occur. It's again through sympatholysis. Let's discuss this further. Now this figure here is an illustration of an arterial vessel cut long ways, so you can see the different layers from the sideways view. As I mentioned earlier, arterial vessels have a smooth muscle wall that you can see right here. And this is made up of course, smooth muscle cells. And the most inner layer is called the endothelium and is made up of endothelial cells. Now pretend this arterial vessel is one that surrounds active contracting muscles during exercise. Now pay careful attention here and take some good notes because not all this information that I'm about to say is indicated directly on this slide. Now when we exercise, we know that this vessel, like all other vessels, are under sympathetic influence, which is going to cause it to constrict. Now during exercise, our actively contracting muscles will release certain substances into the surrounding blood vessels, like this one here. Some of those substances are potent vasodilators, such as a molecule called nitric oxide, or NO for short. The contracting skeletal muscles can produce and secrete nitric oxide into surrounding arterial vessels, causing those specific vessels to undergo a little bit of sympatholysis, meaning the vasodilators like nitric oxide coming out of contracting muscles can override the sympathetic influence on just the surrounding arterial vessels, causing them to vasodilate by relaxing the smooth muscle. And as we illustrated in the previous slide, this will cause a high to low pressure gradient in your arterial system and a subsequent increase in blood flow distribution to the active muscles through the surrounding artery that is dilated. Now, what else happens? Again, pay careful attention to this information. With the increasing blood flow through these dilated arterial vessels surrounding active muscles, there's going to be more friction against the inner endothelial wall. This increased friction due to the increased blood flow through the arterial vessels surrounding active muscles is called shear stress, S-H-E-A-R, stress. Again, the increased blood flow through the initial sympatholysis and dilation of these arterial vessels surrounding active muscles causes shear stress along the inner endothelial wall. This shear stress will stimulate further nitric oxide synthesis, not in the muscle, but in the endothelial cell within those arteries itself. And this is through the activation of the enzyme endothelial nitric oxide synthase, eNOS. This enzyme converts the amino acid L-arginine to nitric oxide. The nitric oxide produced by the endothelial cells will interact with the adjacent outer smooth muscle cells, causing the smooth muscle cells to relax or just simply dilate. Thus, the arterial vessels surrounding active muscles dilate even further, allowing for more blood flow to the active muscles. So to summarize, the initial sympatholysis and thus vasodilation of arterial vessels surrounding active muscle is due to the initial release of vasodilators like nitric oxide from contracting muscles, which causes the arterial smooth muscle in surrounding arterial vessels to relax or vasodilate. Then there is increased blood flow through those specific muscle arteries. Thus, there is increased shear stress along the endothelial wall, stimulating nitric oxide synthesis, which then causes stronger sympatholysis and further vasodilation, thereby increasing muscle blood flow even further. Now, as a side note related to this mechanism of muscle blood flow during exercise, we have a category of performance supplements indicated as nitric oxide boosters, with the claims that it will boost nitric oxide and promote increased muscle blood flow during exercise, and thus increasing performance. Now, I don't want to spend too much time on this here, but if you want further detailed information, please feel free to contact me. I'll be happy to help. Generally speaking, so-called nitric oxide boosters contain specific amino acids that are precursors to nitric oxide synthesis. As I said earlier, nitric oxide is derived from the precursor arginine, and that is usually one of the key ingredients of nitric oxide boosters, as you see here in the form of arginine malate. The other ingredient proposed to boost nitric oxide is citrulline, as you see here as citrulline malate, which is a precursor to arginine. Citrulline is somewhat favored as it is less degraded during digestion compared to arginine. Now here is what the evidence says. When supplementing with arginine or citrulline, there is a very minimal, and I do mean very minimal, increase in nitric oxide levels in the body, which can cause a little bit of vasodilation. But here's the thing, this effect is not just in muscle arteries, but in all arteries. Therefore, it does not result in increased muscle blood flow during exercise, and arginine or citrulline supplements do not enhance performance. They are not regarded at all in the sports science or sports nutrition science communities as effective performance supplements, yet they are still on the market. But here's the ugly side of the supplement industry. Many supplements that are claimed to be nitric oxide boosters, like this one here called nitric oxide shotgun, have very exaggerated and unscientific claims as shown here. Well, the thing is, many do claim that this supplement actually quote-unquote works and that it helps them perform better. And they attribute the effects of this supplement to the claims of increased blood flow to the muscle. Well, if you examine the supplement, The only two ingredients in this entire blend that have anything to do with nitric oxide are arginine and citrulline. And as I mentioned earlier, they do not have a significant performance enhancing effect. What is also included in this supplement is a heavy dose of caffeine, which is a known performance enhancer and can exert immediate stimulatory effects. So when people claim that this supplement quote unquote works, it is not through nitric oxide boosting and increased muscle blood flow. It's likely because they consumed a lot of caffeine right before their workout. Now, I won't say anything specific about this product, which I tested in the lab, since this video is on a public platform and that would be a liability. But I will say that you must be smart about your supplement choices and always be critical of the claims made by industry on any products. This means you need to learn the science, and this will take time. Okay, so moving forward, let's talk about oxygen delivery during exercise, more specifically oxygen delivery to active muscles, since the cardiovascular response as detailed thus far, both in the heart and in the arterial system, to exercise is all predicated on delivering more oxygen to active muscles that require it for ATP production. So the measure of oxygen delivery, more specifically oxygen consumption, is VO2, which is the rate of oxygen consumption. VO2 is a measure of oxygen consumption rate, which in turn reflects oxygen delivery to active tissue and the subsequent uptake of oxygen by the cells of those active tissues. So this is why oxygen consumption, VO2, is also referred to as oxygen uptake or oxygen delivery and uptake. Now, we know that when we exercise, oxygen consumption rate or VO2 increases linearly or proportionally to exercise intensity until you reach maximum intensity, which is where Vo2 max is observed. So how does oxygen consumption rate or VO2 increase during exercise? It is determined by two cardiovascular factors, one being cardiac output or Q, and the other being a variable called Avo2 difference. Now this V here stands for venous. It is not the same VO2 as this VO2, which has a capital V. Just keep that in mind. Avo2 difference stands for arterial minus venous O2 content. Cardiac output, as we know, is a measure of arterial blood flow, so oxygen delivery. While arterial minus venous O2 content, Avo2 difference, is a measure reflecting oxygen uptake by active tissues, particularly active muscle tissues. The relationship between VO2 and these two factors is expressed by the Fick equation, which states VO2 equals cardiac output times Avo2 difference. Again, VO2 is the rate of oxygen consumption, often just simply referred to as oxygen uptake. Either one of these terms are interchangeable. Q equals cardiac output, and Avo2 difference is the difference between arterial oxygen content and venous oxygen content. Now, during exercise, we know that cardiac output increases. Also, as I will explain further in subsequent slides, Avo2 difference increases during exercise. And thus, based on the Fick equation, how would this affect VO2, the oxygen consumption rate? VO2 would obviously increase. We have more oxygen delivery and more oxygen uptake, and therefore oxygen consumption, VO2, would obviously increase. So if I ask a simple question, which of the following factors contribute to an increase in VO2 during exercise, the answers would be an increasing cardiac output and an increase in Avo2 difference. Now let me explain further. Let's focus on the Fick equation above. Here we have oxygen consumption during rest. As indicated moments earlier, VO2 equals cardiac output times Avo2 difference. Now, what are the factors contributing to Q? As we established earlier, the factors are heart rate and stroke volume. Heart rate times stroke volume equals cardiac output. So resting cardiac output is resting heart rate times resting stroke volume. Now, what about arterial O2 content? Now, these values here are per deciliter of blood. So within a deciliter of arterial blood, there is 20 milliliters of oxygen. This is fully oxygenated blood. Please take note of this. Full oxygenation of arterial blood is 20 milliliters of oxygen per deciliter of blood. Now, as the deciliter of arterial blood passes through the capillary network surrounding all the tissues in the body, a total of 5 milliliters of oxygen is taken up by tissues, and coming out the other end of the capillaries into venous return back to the heart is 15 milliliters of oxygen per deciliter of venous blood. This is the venous O2 content. So the Avo2 difference during rest is about 5 milliliters. 5 milliliters was the amount of oxygen taken up by the tissues. Now, how does this change during exercise? We know that cardiac output increases. Why? Because heart rate and stroke volume increases. Now, here's the tricky part. You should know that from rest to 50% Vo2 max intensity, this cardiac output increases because heart rate and stroke volume increases. If the intensity was 50% Vo2 max and above, this increase in cardiac output is only due to heart rate because stroke volume has already maxed out. In any case, cardiac output increases with exercise. This has been already established. Now, what about Avo2 difference? As I said earlier, arterial blood is nearly 100% oxygenated, even during exercise. So this 20 milliliters does not change. So this 20 milliliters of oxygen per deciliter of arterial blood does not change during exercise. Please take careful note of this. Arterial O2 content does not change during exercise. It is already 100% saturated with oxygen. So then what changes? As you can see, venous O2 content decreases down to a maximum of 2 to 5 milliliters per deciliter of blood. What does this indicate compared to rest? As you can see, there is less oxygen coming back to the heart after the arterial blood passes through the capillaries. This means more of this 20 milliliters passing through the capillary network was taken up by active tissues during exercise. So what tissue do you think took up more of that oxygen? Active skeletal muscle. So because active skeletal muscle took up more oxygen out of this 20 milliliters per deciliter of blood, there is less oxygen coming back to the heart via the veins. So during exercise, if arterial O2 stays the same and venous O2 decreases because active muscles are taking up more oxygen, how does Avo2 difference change? It increases. The Avo2 difference was 5 milliliters per deciliter of blood during rest and during exercise, and these numbers are for maximum exercise, FYI. The Avo2 difference increases to about 15 milliliters. Most of that increase is due to active muscles taking up more oxygen. So this is why I said that the Avo2 difference is a reflection of oxygen uptake by active tissues. In this case, active muscle tissues. So if I ask the question, which of the following factors contribute to an increase in Avo2 difference during exercise, the answer is only a decrease in venous O2 content. It is not due to an increase in arterial O2 content. Why? Because again, arterial blood is fully oxygenated, so it can't go any higher than this. Now, based on the Fick equation, if cardiac output and Avo2 difference increases, how does oxygen consumption rate, or VO2, change? It would obviously increase. So if I ask the question, which of the following factors contribute to increases in VO2 from rest to about 50% VO2 max intensity, the possible answers would be an increase in cardiac output, which is due to an increase in heart rate and an increase in stroke volume, as well as an increase in Avo2 difference, which is due to a decrease in venous O2 content. Now, if I asked which of the following factors contribute to increases in VO2 from 50% VO2 max to 100% VO2 max intensity, the answer would be increased cardiac output due to only an increase in heart rate and not stroke volume, and an increase in Avo2 difference, again, due to a decrease in venous O2 content. Now, here we have an animation of Avo2 difference during rest. As you can see in the left side of the heart, in the left atrium, we have 20 milliliters of oxygen per deciliter of blood. So this circle here represents a deciliter of blood, and in it there is 20 milliliters of oxygen. Again, this is fully oxygenated blood. So as the left side of the heart contracts, and more importantly, the left ventricle contracts, the arterial blood will circulate through the systemic arteries. At the peripheral tissues, here we have an example of muscle tissue, but all peripheral tissues, there will be an uptake of about 5 milliliters of this 20 milliliters in the arterial blood through the capillary network. Now coming back, therefore, through venous return, through the veins, will be 15 milliliters of oxygen. And that will return to the right side of the heart through the right atrium, subsequently pumped into the right ventricle, subsequently pumped to the pulmonary system, where this venous blood will be reoxygenated nearly 100% back to 20 milliliters per deciliter of blood. And the cycle will continue. So you have 20 milliliters of O2 in arterial blood, that's the AO2, minus 5 milliliters of O2 taken up by the peripheral tissues, that is your Avo2 difference. You have 15 milliliters of O2 in the venous blood coming back to the right side of the heart. So how does this change during exercise? Again, arterial blood is fully oxygenated. So the 20 milliliters of oxygen within the arterial blood does not increase. It doesn't change. So again, the 20 milliliters per deciliter of arterial blood will be ejected from the left side of the heart, more specifically the left ventricle, and it will circulate through the systemic arteries. As it passes through all the capillary beds, we know that more blood flow is concentrated to active skeletal muscle, which will consume or uptake more oxygen from this 20 milliliters. And therefore, at maximum intensity exercise, almost 15 milliliters of oxygen can be taken up by active tissues, like active skeletal muscles. And what is coming back in venous return is only 5 milliliters of oxygen per deciliter of blood. And this returns to the right side of the heart, and it gets pumped back into the pulmonary system. And even though there is less oxygen in the venous blood compared to rest, the pulmonary system does an efficient job at reoxygenating that blood 100% back to 20 milliliters per deciliter. So you have 20 milliliters of oxygen in arterial blood. That AO2 does not change with exercise, and this again is maximum exercise as an example. You have 15 milliliters of oxygen taken up by the tissue, so the Avo2 difference has increased because active skeletal muscle is taking up more of that oxygen. And coming back in venous return to the heart, you have less oxygen per deciliter blood. You have about 5 milliliters of oxygen in the venous blood. That is your venous O2 content. And like I said, your pulmonary system does a pretty efficient job at reoxygenating that venous blood back to 100% oxygenation, which again is 20 milliliters of oxygen per deciliter of blood. Now here we have an illustration of a Vo2 Max test. And this is something that you would do in lab. Now during the Vo2 Max test, say on a treadmill, you would have stages of increasing intensity until you reach your Vo2 max. Now VO2 would be measured by what we call an indirect calorimeter, or just simply a metabolic cart. You would get hooked up to this system through a facial mask that is interfaced to the device via tubing. And the device will measure oxygen consumption, your VO2. Now, on the x-axis here, you have a scale of exercise intensity as denoted by miles per hour. So this is a treadmill VO2 max test. And on the y-axis, you have VO2, oxygen consumption. Again, also described as oxygen uptake, but this is your VO2. Now, as you go from rest, you start your first stage. And that first stage, let's say, is a 4 mile per hour run. So as you can see, at 4 miles per hour, you know that your cardiac output and your AVO2 difference is going to increase. Your heart is going to pump out more blood per minute, and your muscle cells are active now, and it's going to take up more of that arterial O2. So we know that based on the Fick equation, VO2, oxygen consumption, is going to increase. Now it's going to increase gradually. So as it increases gradually, eventually it's going to reach a VO2 that is sufficient to satisfy the oxygen needs for that 4 mile per hour run. And this is when we hit a plateau in VO2. This is referred to as steady state. So whatever the VO2 is right here where it plateaus, this is the VO2 that is sufficient enough to sustain a 4 mile per hour run. So now what happens if you increase that intensity to 6 miles per hour? Now, obviously, the oxygen demand of the active muscles have increased. So your cardiovascular system is going to respond. Your cardiac output is going to increase, as well as your AVO2 difference, because again, your muscles are more active, it requires more oxygen, therefore it's going to take up more of that oxygen out of that 20 milliliters per deciliter of arterial blood. So based on the Fick equation again, VO2 is going to increase again until it hits a steady state. It hits this plateau right here. So whatever this VO2 is here is sufficient enough to sustain the oxygen demands of this 6 mile per hour run. So now what happens? You increase the intensity again in the next stage. So again, the oxygen demand of the active muscles increases even further. Therefore, the cardiac output is going to increase more and the AVO2 difference is going to increase more. And therefore, your VO2 is going to increase more. Then you see it increase and it hits this plateau again. So whatever the VO2 is here is sufficient enough to sustain the oxygen demands of an 8 mile per hour run. Now let's say in the next stage you increase the intensity to 10 miles per hour, but you don't observe further increases in VO2. It continues to stay at the VO2 that was achieved during the 8 mile per hour run. This means that you have reached your Vo2 max, your maximum oxygen consumption capacity. This means your active muscle cells cannot consume any more oxygen, and this is why your VO2 max is reflective of your aerobic capacity, the maximum aerobic ATP production capacity by your muscles. So this is a very simplistic view of what your Vo2 max test would look like in lab. So here is just a little bit of a flow chart. As exercise intensity increases, we know that the oxygen demand of the active muscles will increase. So your oxygen consumption will increase. Why? Because cardiac output increase, which is a measure of blood flow from your heart. How does cardiac output increase? It increases because of an increase in heart rate and an increase in stroke volume. Now there's an asterisk here because stroke volume increases and contributes to cardiac output increases only up to 50% VO2 max intensity. So that will be somewhere around here, since this will be 50% of your VO2 max. So from here to here, increases in VO2 was due to increases in cardiac output partly, which was due to both an increase in heart rate and stroke volume, since stroke volume maxes out from here onward. Now on the other end, you have an increase in Avo2 difference, not because you have an increase in arterial O2 content. Remember, arterial blood is fully oxygenated. It can't go any higher. This is again 20 milliliters per deciliter of blood. The reason why Avo2 difference increases with exercise is because venous O2 content decreases. Why? Because as exercise intensity increases, your active skeletal muscles are uptaking more of that arterial O2, thus bringing less O2 back to the heart via the veins. So this figure right here should explain the factors that contribute to an increase in oxygen consumption rate with an increase in exercise intensity.